Well, good morning. It's a pleasure today for me to introduce Mary Firestone to give our Distinguished Speakers Lecture. The Distinguished Speaker Lecture series has a monthly seminar that hosts and features an eminent scientist whose work is broadly applicable to Earth Sciences Division work and whose work is outstanding in the field. Of course, Mary satisfies all those criteria. Her work over the past few years at UC Berkeley has involved a whole host of things associated with microbial function, with their microbes' relationships with their environment, competition, with how those processes can be scaled up, importantly for our work, to think about larger scale models. And that's where I first became interested in Mary's work. Some of her work on the nitrogen cycle now informs a lot of the nitrogen cycle components of GCMs. And so I'm excited to hear what she's going to tell us today about the interactions between water and nitrogen and how those processes should be represented in the models. So, Mary? Although that may not be your focus, but that's what I'm going to be listening to. That's what I'm listening to. No, please.
Thank you. Okay, so I'll pin this on and tell me if it's too loud or too soft. I'm going to start by apologizing. I just didn't put it together that today is October 29th and that this is the Earth Sciences seminar for Halloween. And it never dawned on me to bring my witch's hat, and that's probably so it would have been perfect. What an opportunity I have missed. But it is a wonderful opportunity, and thank you for the invitation for coming to come. Soil water is a topic that folks in my group have been working on for a very long time. And so long that, in fact, it probably will take me about 3 hours to summarize everything I want to talk about. I promise I won't do that. I'll try to do it in 50 minutes. But about the first half of my talk is going to be context. And by that, I mean it's going to be old work that's come out of my lab that I think provides the framework that we need for understanding the current questions about microbial processes in water, water control of microbial processes in soil, and then the last half of the talk will be on work that's ongoing now in my lab. So I'm just the mouthpiece. The actual people who did the work and wrote the papers, the authors are listed here. And it's not in temporal or alphabetical order. It's approximately in order that I'm going to talk about the work. And you may recognize some of these names as having been here a long, long time ago, such as Tom Keefe. Oops, he was here a long time ago. But I'll show one piece of his work that is really foundational, I think, for understanding the dynamics of water and soil. And then I'll also be talking about current work in my lab being done by Sarah Placella, Catherine Osborne, and Romain Bernard. Okay, so how do bacteria and Archaea in soil experience water? And I like this image. And had I really thought about what day it was, I would have come dressed up as a bacteria to try to give you a perspective, which would have been better than a witch's hat. But we're going to start with a ridiculously simple model, and that is these are bacterial cells growing on a mineral surface, and this is the water film that's covering them. And what can we learn just starting with this simple model? Well, in California, our Mediterranean grasslands dry out in the summer, and they dry out really dry. And so we're going to start, these are two field sites that we work in. One is down near Santa Barbara, Sedgwick, and one is up between Marysville and Grass Valley. It's called Sierra Field Station. And the third site that I'm going to talk about is Hopland, which is about 3 hours north of here near the Fetzer wineries. So these are sites that, as all California annual grasslands do, dry out substantially as the spring progresses and as we move through summer. So we're going to quickly think through what happens as soil dries. The water films in soil become thin very rapidly. How rapidly? So this is water film thickness in microns. And let's say it stopped raining about 24 hours ago. And we went out and we took our little micrometer and we measured the thickness of the water film around soil particles. And we would find that about 24 hours after a rainfall event against gravitational pull, the films would be about 4 microns thick. And that would give us a water potential of about 0.03 megapascal, or minus 1/3 bar. That's almost by definition 24 hours after a rainfall event. Well, as soil continues to dry, eventually we will get to about 10 molecules of water thick. And at that point, the traditional is that plants go into a permanent wilt. Well, you know, some temperate plants will go into a wilt and will not be able to recover. That's a kind of an old saw. That's minus 15 bars, or minus 1.5 megapascal. And in August, in a California annual grassland, it will require a pickaxe to be able to sample the soil, and we will be well below minus 10 megapascal. So we're now getting down to a couple of molecules thick on surfaces. Okay. One of my graduate students asked me to translate the units, so I'll do a quick translation. Water potential units are in basically in units of pressure. And 1 bar can be expressed in atmospheres, but the units that we most commonly use are megapascal. And the translation between a bar, if you think in bars, or kilopascal, and megapascal. I think in terms of megapascals, but I'll try to translate as we go. Now, in soil, these water potential, or psi values, are generally negative. And that means that it's all expressed relative to pure free water, that the water potential of water in soil is less than pure free water. Okay. So going back to this, this would be minus 1/3 bar, this would be minus 15 bars, and this would be minus 100 bars. And in fact, we've now done some work recently showing that California grasslands in August commonly get down well below minus 30 megapascal, and that's minus 300 bars. Okay, as soil dries, water film thickness decreases rapidly. If we look at the thicknesses of those water films, we can readily see that limitations of motility of protozoa and bacteria begin to occur relatively rapidly at about minus 0.1 megapascal. But at least as important is the fact that limitations of diffusion due to the thicknesses of the water film become critically important very quickly. If we look at the rate of solute diffusion, could be carbon substrate, could be ammonium, could be a simple ion of some sort. It is dependent, the rate of diffusion in soil solution is a function of the water content, theta, to the 3rd power. So the diffusional characteristics of soil are extraordinarily dependent upon soil water content, extraordinarily as in to the 3rd power. Okay, so what we're going to think about is how diffusion or potentially starvation limits microbial processes as soil dries. As soil dries, the soil water potential, or the energy availability of the water, declines. Also, energetic availability to microbes of water is reduced, and as soil dries, the soluble salts, or the salts that are remaining in solution, concentrate, and we get specific ion toxicity resulting from concentration of salts, particularly sodium and chloride. Okay, as soil dries, microbial activity is reduced. We know commonly rates of processes decline as soil dries. It can be due to a diffusional limitation of substrate supply due to the decreasing thickness of water films. It can be due to physiological impairment due to water stress, to the energetic availability of the water. It can be due to specific ion toxicity, or a combination of all of these. So, in an attempt to determine how much is due to, as soil dries, microbial processes decrease in their rates, and how much of that decrease is due to a diffusional limitation of substrate availability, and how much of it is due to physiological impairment, okay, due to low water potential and specific ion toxicity. So we're going to test a very simple microbial process where we can control the supply of the substrate independent of the water film. That is, we can supply the substrate through the gas phase, and then we can measure the rate of the process over time. So the process we're going to look at is nitrification. Okay, it's one of our favorite processes. Two critical substrates, oxygen through the gas phase, and we can supply the other substrate as ammonia through the gas phase. So independent of the amount of water, we can control the substrate availability. And we readily, by pool dilution, can measure the gross rates of these processes. So this is kind of a complicated, and this is old, 1995, John Stark's dissertation work. Okay, so what we've got here is a wet soil, free water, 0. The rate of nitrification, which is shown on this axis, is maximum. Starts up right here. Now this one is drying down. So we're decreasing the water potential substantially, minus 1 megapascal, 10 bars, down to 30 bars due to desiccation. And we measure the rate of nitrification, the gross rate of nitrification, and it declines as we dry. And then this top line, we're controlling the water potential by addition of K2SO4. So this is being controlled by a solute. And in this case, we're supplying ammonium, excess ammonium, so there's not a substrate limitation. And so what we're going to do is try to assess how important diffusional limitation to this process is by looking at the difference between this line and this line. In this line, water film thickness and diffusional limitation is in action. And in this line, it is not. So if it's due to diffusional limitation, if we go into these flasks and add ammonia gas, such that there is no longer a diffusional limitation, the rates of these processes should approach this line. Okay, so we do that. We go in and add ammonia. And what we find is not only, let's look at these, do the rates of nitrification in these very, very dry soils. Out here we're approaching minus 25 bars. Not only do they approach that in the K2SO4 poised soil, but in fact they get higher. So probably we're getting some specific ion toxicity here, and in fact we can not only recover those rates. So as we dry down, a significant part of the limitation of nitrification is due to diffusional limitation, and in fact we can estimate how important diffusional limitation is versus physiological impairment, which is what we're doing here. Here's 0. This is the wet system. The fraction of decline, there's no decline, it's 1. Full speed. Now, as we begin to dry the system down, we find that substrate limitation is extraordinarily important at the higher water contents. As we dry the system down, we get here to about a 50-50 point at about minus 0.7 megapascals. At about minus 0.7 megapascals, the decline in the nitrification rate is due about 50% due to diffusional limitation and about 50% due to physiological impairment. As we get drier, it becomes dominated by what I call dehydration effects or physiological impairment. So we know as soil dries that up here, 24 hours after a rainfall event, up here for the first few days, we're getting motility limitation. And the wetter soils, as it begins to dry, we're getting diffusional limitation. And then about the time we get down here a little bit above, where plants minus 15 bars tend to go into a permanent wilt, we're getting substantially, almost totally, physiological impairment of the microbes present. Okay, so we've got this simple model of water films. But how do microbes really experience water in soil? What does the world really look like? Well, obviously way more complicated than this also. But minimally, most bacteria live in exopolysaccharide matrices in soil, EPS. What does that do? These are dominantly water, like 98%, 95% water. Okay, so this is a picture of a pet Pseudomonas from soil growing on sand at a relatively high, relatively moist conditions, minus 2.5 bars. And here's one growing, the same bug, at minus 1.0 megapascal. And what we're seeing with this Pseudomonas, and we've seen with a number of organisms now, is as you grow them and decrease the water potential of their environment, they will make more and more EPS. It seems to be made in response to desiccation. Okay, so it's not surprising. They're controlling their own microhabitat. Here's another picture of a Rhodococcus, and this is out of Trish Holden's lab at UCSB, done by one of her students. And I just think it's a pretty picture. We've got a lot of pretty pictures too, but between two sand grains. So what are the roles of EPS in soil in terms of water? If the microhabitat of the organisms is in fact an EPS matrix, is the water content, that's theta, of that habitat higher even at a given water potential, psi? Okay. EPS cannot change the energetic status of the water. It cannot change. It's going to come to equilibrium. But at a given water potential, it can change the water content. Okay? So, two questions. Is the water content of the microhabitat higher when microbes are embedded in EPS? That's important because we know that diffusional supply of nutrients to organisms is dependent on the water content, theta. The physiological impairment is dependent on the energetic status of the water, and that's psi, water potential. So if the water content is higher in a microhabitat, will that allow continued microbial activity at low water potentials? As soil dries, if you're embedded in an EPS matrix, can you continue to function under drier and drier conditions? And does EPS modulate or slow down the rate of change of water potential that microbes experience? Now, drying generally occurs relatively slowly in soils, and microbes have an opportunity for physiological response to drying. And there's a lot of work on the physiological response to desiccation. Is it possible that it also modulates the rate at which microbes experience wet up? And that's a good question, and this is another cassette of about 10 minutes that I took out, or else I would be talking for 3 hours. I will simply tell you the answers to these questions. This is Emily Roberson's work, published in AEM. Is the water content of the microhabitat higher in extracellular polysaccharide? Yep, not surprising. At a given water potential in soil compared to say, pure sand compared to sand plus EPS, the water content will be about 100% higher in the presence of EPS. Does it allow microbial activity to continue at low water content, at low water potentials? Answer, yes. I mean, it can only do so much, but it enhances the capacity to continue. And does EPS modulate the rate of water potential change? Answer, yes. Okay, so we've talked about slow drying and microbial, what it looks like under static conditions. But water is not static in soil. It keeps drying down and wetting up. And this is an old picture. It was old when I pulled it out, of Hilgardia. It's data from Sierra Field Station. And what I want you to look at are the the lines. This is time, wet season, November, December, January, February. We're in California, it's raining. And now we're getting into spring and we're having these little dry periods. And as it dries down 4 to 8 centimeters, which are the lines, the water potential begins to decline, okay? So let's hear about minus 20 bars or minus 2.0 megapascal. We're having a drying event in May. We're getting down to minus 2 megapascal, and then it rains, and we get a wet up event. And so, in fact, we use this data to assume that even in the spring, microbes are experiencing water potential upshocks or wetting events on the order of 2 megapascal. So we went in, and this is the old data from Tom Keith I was talking about. And he looked at soil, and he said, we looked at dry soil. The starting here was about minus 2.8 megapascal, about what you might have seen in the spring in a California annual grassland. And then we wetted it up. We did a rainfall on that soil. The starting total microbial biomass under these conditions was about 1 meg of carbon per gram of soil. And the wetting event released about 17% of the biomass carbon. With a larger wetting event, looks like a pretty big one, 6.9 megapascal, or 69 bars. Well, we're going to find out that in the fall, when it starts raining in the California annual grassland, we get upshocks that are substantially larger than this. But let's say this, we're looking towards fall, end of the long dry season, rainfall event. And we're liberating, wow, a third of the biomass carbon. So with rainfall events after long periods of drought, microbial biomass goes on the market. Why? So to answer that question, we looked at 4 cultures isolated from grassland soils, 2 gram negatives and 2 gram positives. Since we've got 4 bacteria, in fact, we can make global statements. That was a joke. We did, however, it's generally thought, even then, that gram-positives responded quite differently to water than did gram-negatives. So we were trying to get 2 gram-positive soil bugs and 2 gram-negatives. So first, let's look at release of amino acids from the cells after the cells are exposed to a dilution event of about 2 megapascal. So that's about what we were looking at in soil. It's about what we see in May in a California grassland. So what percentage of the amino acids are released? The 2 pseudomonads run between 10-25% of the amino acids are released by this 2 megapascal upshock. Oh, the gram-positives, in fact, are much more resistant to the water potential upshock. Now, this column was toluenized cells, and if you know, toluene dissolves the membrane. So actually what we were trying to look at is how much would go on the market if we literally destroyed the membrane. And so this was kind of the control of what could we measure coming out of the soils in terms of amino acids. So if amino acids were being concentrated as compatible solutes, what percentage would go on the market? What about neutral sugars and protein? Again, the same dilution stress, about the same percentage of neutral sugars, which makes sense. Small molecular weight amino acids, small molecular weight neutral sugars. And again, the gram-positives were very resistant to dilution. What happens when microbes experience, after they're physiologically adapted to a given water potential, they've osmoregulated, and you wet them up? It's potentially the opposite of plasmolysis. Plasmolysis is when the cell membranes pull away from the wall. Plasmoptosis is when water gushes in, the pressure becomes too great, and the cells pop. Well, that is known to happen with lichens on rock surfaces when it rains. And so we were interested in where these cells were actually popping or whether the membranes were becoming selectively permeable and they were losing the smaller molecular weight compounds. So that's what the protein release is supposed to show. Proteins are large molecular weight compounds, and if the cells were popping, plasmophysis. They would go on the market, would see the proteins and the DNA released, and we didn't. These cells seem not to be exploding, but rather they're gushing out a portion of their concentrated compatible solids. Okay, makes sense? So, evolutionarily, these microbes have been indigenous to soil for a long, long time. The question was, interesting to ask, are soil microorganisms highly adapted to tolerate wetting events? Because at least in a semi-arid ecosystem, they see it all the time. So here's our two bugs down here. Here's our Streptomyces and P. fluorescens from soil. And what we really did was total release of soluble cell sap. The best comparisons we could find at that point in the literature. Here's an E. coli from the intestine, a bug from a marine indigenous bug. And when they have a 1 megapascal dilution stress, they really lose it. They are not at all tolerant of water potential upshot or dilution. An aquatic, freshwater aquatic organism is more tolerant, but the organisms found indigenous to the intertidal zone, where they see salinity fluctuations on a regular basis, we found to be just about as tolerant to water potential wetting or dilution events as were the organisms indigenous to soil. Okay. So, the first rainfalls following extended hot, dry periods in arid and semi-arid ecosystems cause high fluxes of CO2. And this, we were talking about this before, this is work that's come out of Dennis Baldaki's lab in particular. If you look at his tower data in California and Mediterranean ecosystems, about 80% of the CO2 fluxes occur within 24 hours of a rainfall event. It is an incredibly pulsy system. It is driven by rainfall events. Okay, here we're looking at wet-ups in 3 California annual grasslands, the Hopland, the northern site, Sedgwick down by Santa Barbara, and this is the beginning of work currently being done by Catherine Osborne and Romain Bernard, and all of this is as yet unpublished and in fact in process. So here are our CO2 pulses in response to wet-up events in field soils. The first rainfall after dry periods in arid and semi-arid systems cause high fluxes of CO2. Our microbial ecology questions were, how do bacterial physiological states change during the dry down that happens over the summer? Are groups within the soil microbial community particularly adapted to respond rapidly to wet-up events? What metabolic capacities turn on with a wet-up at the end of a long dry period, and how fast? And in a global change context, how would changing patterns of precipitation be expected to change the response, the compositions of the community, and the physiological characteristics and responses of the community? Okay, so the process, is Catherine and Romain doing a project that's ongoing, looking at literally the answers to these questions. So here is a rainfall pattern in California, a soil water potential pattern. It stopped raining in April, it begins to dry down, and it stays dry until September. Now it's going to start raining in September, wet up in September. So that is an approximately average. There is no such thing as average in California. We all know that, but if we pretended there was, this is what an average dry down pattern would look like in September, and then would get a wet up. So what they imposed was an extended rainy period, which is one of the GCM predictions for California, that in fact the rains will continue later in the year, basically until early summer, and that's what happens here. Rainfall, the dry down is delayed, and then we also compared it to a third treatment in which there's no dry down, and that is we kept the soil moist all summer. And then Catherine and Roman came in and sampled 3 times at about here, 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 and here. Now this is to look at the physiological responses of the communities indigenous to the soil. Okay, under controlled conditions in soil from a California grassland brought back to the greenhouse, can we identify components of the microbial community that have an enhanced capacity to respond rapidly to wet-up events? So, have there evolved adaptations in the community that they know as it dries down, that if they are hunkered down at the starting line with lots of ribosomal RNA and ready to start quickly, that there will be a wet-up event and that there will be a lot of carbon on the market? Is there a strategy? Do microbes actually carry strategies for quick response? We want to look at the turn-on of key drought survival genes during the dry down. And we also want to look at specific genes that would control the capacity of the indigenous community to utilize carbon that comes on the market quickly with wet-up. So this is a greenhouse experiment, and this is a field experiment. 3 California grasslands, basically using field studies to see if the types of insights we're seeing in a controlled greenhouse really do, in fact, extrapolate to the field. Are there components of the bacterial community with the capacity to respond quickly? This work is being done by cDNA analysis of the RNA content, ribosomal RNA content of the indigenous communities during the dry down using the G3 phylochips out of the Gary Anderson's lab. Over time and desiccation, what drought survival genes turn on, and what kinds of capacity to use potential carbon substrates can we see? We're doing this by qPCR of bacterial drought-resistant strategies. And basically, Catherine has primers for sporulation out of firmicutes and out of streptomyces. She has EPS producing genes, primers for EPS producing genes out of 3 different groups of bacteria. And she has a range of compatible solute producing genes. So what we're trying to do is kind of you can't assess or test everything. These are selected primers that are available that we can use to try to follow response to desiccation and position, if you will, positioning at the end of the summer in terms of being able to use carbon that comes available. Some of this data, well, we have some of this data, but Catherine embargoed it because she says it's not ready for prime time, and she's right, it isn't. But I will tell you just a couple of things about the qPCR of these genes during dry down. Okay, and then also we're going to determine whether what we're seeing in the controlled environment is applicable to the field. And here's the field component. We're looking at 3 sites: Hopland grassland up near off 101, about 3 hours north of here; Sierra foothills between Marysville and Grass Valley; and Sedgwick down near Santa Barbara. Here's Sedgwick, Hopland, Sierra. And here's the cumulative precipitation over this past year, the year we put the cores in, and it was very well-behaved precipitation, which is unusual in California. Am I in your way, Bill? So what we have is, this is the far south one. We expect it to be the driest. And here it is now. Precipitation, only beginning in January. It's raining, it's raining, it's raining. And now it stops raining. Here's the Sierra field station, Sierra foothills. And then the Hopland field station here has the greatest precipitation, cumulative precipitation. And so we put cores in, I should say, Romain and Catherine put cores in here when it was still wet. Cores were pulled here, here, and here after a long dry period to look at basically the physiological state of the organisms in the field as well as wet up responses. Okay, now we're back to the greenhouse experiment. These are controlled. The greenhouse experiment looks like that. Okay, there they are. Cores taken from the field intact, come back to the greenhouse, and we control the dry down. And the dry down, we have our long field-like dry down here. We have the extended wet period, which is in black. Whoops, it's wet, it's wet, it's wet. Now it begins to dry down. And then we have the cores that are watered once a week. So they're very flashy here, but they stay at a much higher, this is the weight of the PVC, but it represents the water content. Okay. And what I want you to look at over here is the water potential, which is what really matters in the soil. And this is over time. So here we go. Here's our T0 in April. Here is our simulated no more rainfall. And we get water potential decline down to about minus 30 megapascal. Well, if you've worked in water and soil in a long time, minus 30 megapascal is shocking. That's minus 300 bars. And it's very dry. We know it takes a pickaxe to get it. And this is really how dry California grasslands become in the summer. Here's the one with the extended wet period. Now we stop watering it, and it quickly comes down. By the end of the summer, it's just about down to the same water potential as our extended dry. And here's the wet, the one we kept watering over the summer. So the water potential stays pretty high. Okay, so those are the patterns of water potential over the summer. Okay. Right, and then we come in 3 times and test it. Okay, now, T0, and by this one, we're at September, we're at the very end of the summer, and this is total DNA extracted from the soils, and there's no statistically significant differences in the amount of DNA. It's kind of frozen in place. What surprised us was if we look at the soil that begins to dry right away, we quickly begin to be able to extract significantly more RNA from the soil. And to reiterate that, the sample that had the black one, the extended wet, follows the green, blue, and then as it begins to dry, it basically begins to do the same thing. We begin to be able to pull more RNA, extract more total RNA by pico-green out of this soil. So this was unexpected, and we assumed out the starting gate that it was methodological, that when soil got really, really dry, we were inactivating the RNA ACEs, or for some reason we were much more effective at being able to extract RNA under these conditions. So Catherine has been working on controls for about 3 weeks now, adding RNA under dry conditions, extracting it, trying to find a methodological physical explanation for these enhanced amounts of RNA. And to date, it's still biological, but she has one more control to run. So at this point, we're operating under the assumption that this is real, that in fact there is more RNA frozen, more RNA has been produced and is now frozen in place and would be produced during the dry down. And in fact, it's not crazy. There's in fact a number of possible mechanisms that could explain that, which I won't talk about yet, unless you want me to. One thing that is interesting, looking at 16S transcripts, is that there's no shift in activity between bacteria and archaea. So looking at archaeal ribosomal transcripts and bacterial, they both seem to be impacted, if you will, by desiccation, about the same. And here's the CO2 fluxes from, let's look first at the wet, the one that maintained was pretty wet. for the whole period of time. Very little CO2. The driest one, the one that got driest, has the greatest CO2 flux, and the extended wet period is intermediate. Quickly, are these patterns relevant to other trace gases, specifically nitrous oxide? And this work is Sarah Placella's dissertation work. And what she shows here, this data is from Sedgwick soil, and it shows background rates of CO2 and N2O in red, CO2 in blue, with wet up. Now, this, for long story, is a log scale of time. I'm going to tell you why in just a minute. But what we do see is that we do see a major pulse of N2O in response to wet up as well as CO2. The pattern's a little different. The time frame is a little different. The organisms responsible are a little different. What is the origin? Sarah's thesis, chapter 2, is on what is the origin of nitrous oxide emissions resulting from wet up, and on what time scale do the organisms potentially responsible for the N2O respond to wet up, and how does that compare to heterotropes? So she sampled Sedgwick, brought it into the lab, composited to do a reduced variability, and then she wet up 10 mils of soil, 10 mils of water to 40 grams of field wet soil. Field wet is a misnomer. It's 4% water content minus 22 megapascals. It's very, very dry. It takes a pickaxe to sample it. She wets it up. She looks at these things at before wetting, 15 minutes, 30 minutes, 1 hour, 3 hours, 9 hours, 24, 72 hours. The only way you can get that time frame on the same graph is to do a log of the time. And that's why everything you look at for the next couple of slides is going to be on a log scale of time. She looked at CO2 and N2O fluxes, which you just saw. And then she looked at transcripts of the ammonium oxidation genes in bacteria and Archaea, the nitrite oxidation gene in nitrifying bacteria, and then denitrification genes, NERK for nitrite reduction, NOS-Z for N2O reduction. So we're looking at the gene, the transcripts, the expression of the gene responsible for ammonium oxidation. We're going to look at, again, the expression of the gene, transcription of the gene responsible for nitrite oxidation, and then we're going to look at nitrite reductase and N2O reductase. We're going to start with the nitrifiers because that's the more interesting data. We're looking at the transcription of the genetic capacity to make these enzymes. So here is bacterial AMO transcripts over time, and this is a log scale. Here's 1 hour. Here's pre-wet up transcripts. Here's 1 hour, 9 hours. The transcript production peaks at 9 hours after wet up and then begins to decline. Bacterial AMO transcripts. The blue is now archaeal AMO transcripts, and that is, okay, so how fast are the archaeal nitrifiers stepping up? A little slower, not very much, pattern very similar to the bacterial nitrifiers. And finally, the green is NXRA, or nitrite oxidation. So that will be the enzyme, transcription of the enzyme that will take the product of the ammonium oxidizers and oxidize it from nitrite to nitrate. So these are the transcripts of the nitrite oxidizers. Okay, so it lags behind significantly. It lags behind, but it also revs up relatively rapidly. Now, this is the first point that the bacterial AMO increase is significant. The first point, the NXRA is significant, and the first point, I think, that the archaeal AMO is significant. So what we're seeing is a turn-on of transcription of these enzymes in a period of about 1 to 9 hours. And this is much faster than these organisms grow. Sarah also did DNA on these organisms, and there's no change. in the number of gene copies. They're not growing. They're transcribing as a way, in response to a wet-up event, as a way of responding to the wet-up event. Okay, these patterns overlay. This is ammonium accumulation in soil. This is nitrate accumulation in soil. And they are very logical and very consistent with the transcripts. What we see is that the ammonium oxidation transcript abundance has an R-squared of 0.82 when plotted against the soil ammonium concentration. Now that doesn't sound very surprising at first glance until you think about it a little bit. Because what it's really saying is that it's not, we've always known that nitrifiers grow in response to ammonium availability, but that they induce synthesis de novo of enzymes in response of increasing ammonium availability is not something that you will find in the pure culture nitrifying literature. Okay, so nitrifiers are definitely responding to the wet up. Denitrifiers, we look at NERK, nitrite reductase, and NOS-D, and that's right here. We can find no transcription of the denitrification genes in response to the wet-up event. It appears that it's a nitrifier activity that is consistent with the consumption of ammonium, the production of nitrate, and the production of N2O. Denitrifiers do not show activity. The AMO transcripts explain about 87% of the variability of the nitrous oxide fluxes. Nitrous oxide emissions following wet up are due to ammonium oxidizing bacteria increasing their activity. We are unable to detect any increased activity of, in fact, any activity, it's not too surprising, these are semi-arid grassland soils, of denitrifiers. Transcripts in this system are serving as the control point following wet up. Transcriptional control happens very quickly, within an hour, in many systems, and controls the rate of ammonium oxidation. Now, is this surprising? Well, it's one of those things that when you think about it in retrospect, it doesn't sound too surprising, but it's something we really haven't thought about. And what this is saying is that the microbial control of a very important atmospherically reactive trace gas is dominated by transcriptional response of the extant community. How do microbial communities responsible for denitrification, for N2O, maybe for CO2 production, and maybe even in some systems for methane oxidation and methane production? In many systems, these trace gas productions are very pulsy. They are episodic, and they occur relatively rapidly in response to changing environmental conditions. So rapidly that we're not seeing microbial growth in response to changing environmental conditions. We're seeing microbial turn-on in response to changing environmental conditions. We're seeing microbial transcription of enzymatic capacity in response to changing environmental conditions. And this is a different way of looking at microbial control of process, of episodic process, but particularly atmospherically reactive trace gases. Was telling Bill, I was talking with Leif Clemetson, who's microbial ecologist gone to the dark side. He's now a tower jock who measures trace gas production in Sweden. And we were talking about the fact he spent years trying to put microbial growth models, particularly for denitrification, into DNDC. And he says that eventually they've realized it just doesn't do any good. And that what they in fact need for processes that are very episodic is some way of getting a handle on microbial response to rapidly changing environmental conditions, such as, in Mediterranean ecosystems, wet-up events. So this is kind of a first look or a first thought about how can you begin to describe the amount of activity you can get due to an extant microbial community responding to a changing environmental condition. So that's actually ultimately what Sarah will be writing about. Right, Sarah? Okay, and I've almost talked, what time did I start? Pardon? So, 3 minutes? Okay. Okay. Well, anyway, so actually the rhizosphere is also a topic that's long been of great interest. And overlapping what's happening in the root environment with water potential relations is a really interesting question. We know that the root environment is an environment in which water potential changes relatively rapidly. Okay, this is old stuff from the literature. This is a wet soil at day or night, and this shows water potential stays high in a wet soil. But if you go out into a California semi-arid or a slightly dry soil and look at what happens, now this is out in the bulk soil, and now we're going to move towards the root, and we're going to look at the water potential during the day when the root is actively evapotranspiring. What you find is that the root pulls down the water potential of the soil immediately adjacent to the root, shown here. Oops. The water potential declines during the day, and that as the evapotranspiration stops at night, the water potential resets. So we have a 24-hour water potential fluctuation, or cycle, if you will, that occurs in a root environment in a relatively dry soil with an evapotranspiring plant. Okay, so this is a question that Romain Bernard has been interested in a long time. And ultimately, this was the basis of a Marie Curie project that he came here over almost 2 years ago now to work on. Do water potential fluctuations in rhizosphere soil impact the nutrient availability to plants? We know water potential fluctuations do a lot of things to communities. Can they be in part responsible for pulses of nitrogen mineralization? And or are the microbes indigenous to a rhizosphere community even more highly adapted to tolerate water potential fluctuation? So I know I don't have time, and besides, Romain doesn't have any data yet. I do want to show you the experimental setup that he actually developed with Rohit, who's not here, but and Don Herman, and this is Romain. Okay, and that's greenhouse data, which we're not going. From a soil microbes perspective, particularly in a semi-arid ecosystem or an arid ecosystem or a system that experiences significant drought, life is largely about water. In a dry soil matrix, water availability and water dynamics, the water content and the energetic availability of the water, which are two different measures, control the availability of substrate and nutrients to the indigenous microbes. They control the physiological state of the microbes that are present. Ultimately, it can have a controlling impact or significant impact on the production of atmospherically reactive trace gases such as nitrous oxide, CO2, and methane. And it does impact nitrogen cycling, particularly nitrogen mineralization and nutrient availability in the soil immediately adjacent to the root. And so that is the final conclusion. This is California annual grassland. And I realized, is there an animal there? Is there? Is it chipmunk? What is it? Have you never seen that before? Sarah gave me this picture. I was looking at it. Maybe it's a dog. Anyway, so this is your average California annual grassland. It's a tough place to work, but we never have any trouble getting volunteers to go to the field. So, any questions? Yes?
Do we know how much the high potential, the high negative pressure impact the thermodynamics of the system? In terms of free energy of reactions, and those potential gets really high, and I'm wondering how that can impact the.
I don't know the answer to that. Can you help me with the answer to that? We are talking about the activity of the water, so any reaction that is occurring in solution, solution here being used loosely, because we may be talking about molecules of water thick. It's a very interesting question. I have no idea.
So I'm wondering if that would affect some of the thermodynamic drives for various microbes. I mean, I don't know.
Could it?
Our activities are going to be very
Okay.
low.
Could it affect could it affect activation energies for enzyme reactions? Is that what you're suggesting? Anybody have any good ideas? Yes. Would you?
Well, this is perhaps a related question. To what extent does the nature of the substrate affect the resistance of bacteria to water stress? For example, for nitrogen-fixing bacteria, where they don't use a hydrated substrate, they use nitrogen.
Okay. So we've actually worked on that quite a bit. So the point of it is that if, in fact, your substrate is coming into the gas phase, then being dried out in a thin water film, in fact, makes that substrate more available. And Trish Holden, a graduate student in my lab, did a lot of modeling on reaction diffusion models, basically diffusion into the water film, dependent on water film thickness, of VOCs, volatile organic carbon compounds, particularly including toluene. And the answer is yes. The answer is, to the degree, here's the water film, to the degree that the toluene coming in from vapor phase is becoming more available, desiccation accelerates the rate. But when you get to the point that you're beginning to get of toluene utilization, when you're beginning to get energetic availability of water that's so reduced that you're getting physiological stress on the bugs that are doing the reaction, it begins to slow the rate down. But for something like nitrogen fixation, where your substrate is coming in from the atmosphere, but man, you've got to get that carbon, you've got to get that energy from someplace. So maybe if you're sitting on a root and it's being piped in right away, then you might be less sensitive to desiccation than a microbe that was after a large molecular weight carbon compound that now has to diffuse in through a thin water film. Better you're a nitrifier and you're looking for ammonium, a relatively small compound, and CO2. I had something else to say, but I can't remember. I'm sorry. Yeah, but it's an interesting way of looking at it. Okay, somebody.
So just thinking about kind of ecosystem scale fluxes, so CO2 measured on the dark side on the tower. And so how would, for example, if we look at fungal metabolism versus bacterial metabolism, and so for example, in this system here, what fraction of the metabolic fluxes are really controlled by fungal metabolism? And are there similar relationships between, for example, the response of fungal populations to web?
Oh, I don't know. Okay, so are there similar relations of, okay, so if we're talking about decomposition, you've got to play fungi in a major way. If you're talking about nitrification, denitrification, we're dominantly bacterial. If you're talking about nitrous oxide or methane, again, we can talk about bacteria and Archaea, and at least in terms of the immediate reaction, and have some confidence. But if we're talking about decomposition processes and total CO2, you've got to get fungi into the model for sure. So fungi experience water in terms of energetic availability of water, just like every other life forms, plants, bacteria, Archaea, fungi. So in terms of psi, water potential relations, stress, specific ion toxicity, I think the reaction, and there's work out of Griffin that shows the reaction is very similar. In terms of substrate supply. Bacteria are stuck in the water film waiting for the substrates to diffuse. Okay, so they have flagella. In theory, they could swim towards, and if it's wet enough, they might swim towards, but it has to be pretty wet, okay, for them to swim towards the substrate. Fungi have these hyphae that go aerial. So now you're no longer relegated to water films. In terms of metabolism, they probably are, but they can cross air gaps. And so that is a major way, in theory, of getting around diffusional limitation. They can go to the substrate, particularly macromolecular carbon substrates. So yes, the fungal story is different.
Like, for example, this system here, if you were to look at, so let's just take off autotrophic respiration and look at just heterotrophic. Any idea, like, how we would partition that? Curious. And in, like, for example, this system.
Okay.
Fungal and bacterial.
Autotrophic being root respiration?
No, just excluding that. Just look
Okay.
at heterotrophic fluxes partition between bacterial and fungal.
Mm-hmm.
Just curious if there's any work done on partitioning those two in any system for this system.
Actually, yes, there is, and I don't, off the top of my head, I don't have it. There are a reasonable number of studies using broad spectrum fungal inhibitors and broad spectrum bacterial inhibitors attempting to identify the source of CO2. Now, those tend to be in relatively water constant, dry, wet, medium environments. I've never seen that done in a wet up. Have you? No? But that's an interesting question. Interesting question. Okay, so that's one thing. So I talked about what happens to the bacteria in the Archaea during the dry down. And we just realized last week that we really have to look at what's happening to the fungal component also. And so that's we're just actually starting to do. JP.
I don't know if it was a month or 3 months ago, but we were sitting in this room and listening to
Josh.
and
Yes, which is what inspired me to talk about water. Go ahead. Yes.
he was talking about a somewhat similar topic, and he told us that during this dry season, you actually get an increase in microbial ionosis. And I don't see that your data necessarily contradicts that anyway, but it intuitively
Makes no sense.
seems, yeah.
Okay. So actually, if we start seeing this increase in RNA, but that's not the same as an increase in microbial biomass. And if you ask Josh, how did you get the increase in microbial biomass? It's by chloroform fumigation, direct extraction. And so you got to take it with a grain of salt, or you've got to take it with a beaker of chloroform.
So let's get away from that, because maybe it's a non-issue.
Okay.
But, so the water potential measurements are a very bulk analysis. And you showed with your EPS that you could start to get patchy distribution where you could actually have growth. You will certainly continue that.
Wait, you get patchy distribution of water content, water. You don't, over time, get a patchy distribution of water potential. The energetic availability of the water comes to equilibrium. Now, polysaccharides slow down that equilibrium maybe by a day or two, but they're not going to change the equilibrium. The energetic availability of the water in the EPS is still going to be minus 20 megapascal. And that's what Trish Holden's work looks at, what happens to the EPS, which is a hydrated matrix, when you dry it down, and it kind of goes away. But the water content at a given water potential will commonly be about 400, 500% higher in the EPS. So it is possible at those intermediate water potentials that you've got enough water present to still have diffusional supply. And if your polysaccharide matrices are also carrying your extracellular enzymes, they're retaining water, they're carrying your extracellular decomposition enzymes, then you've kind of set up a sweet system that you can still have decomposition and diffusion. Decomposition, product production, and diffusion. And this doesn't even go into quorum sensing molecules, which are a whole nother story, which I eliminated, yes. Owen.
So you showed that, or
I'm going for the tough questions here. Go.
Sarah's data has shown that we can relate the AOA transcripts very well to N2O emissions, and you don't see a similar relationship between gene copies, so there's no growth. That's pretty clear.
Yeah.
But if you compare across different sites, maybe you see the same response. But is the magnitude of N2O emissions controlled by the standing biomass of the isotopes?
So,
The number of catalysts?
okay. So, Bill and I started to have this conversation right before. And so, why do you want to know about the size of the standing population? Because in the end, that's going to define your Vmax, isn't it? So you've got a standing community that has characteristics of response characteristics due to the speed at which they can transcribe and turn on these critical processes. The patterns that Sarah found in Hopland, Northern California, and Sedgwick, Southern California, were staggeringly similar. I mean, so now we've got 2 California annual grasslands showing the same pattern. So clearly, again, it's true of the entire globe. But at least it was satisfying that it happened twice. Okay. So, but is the absolute magnitude of the N2O different, and is that due to the number of ammonium oxidizers present? And I don't know the answer to that. It's a good question. Yes.
So I was thinking about, what happens when you start integrating this in depth? I mean, clearly there's depth gradients to water content and water potential, above the capillary fringe. And I have no idea in these types of grassland environments, are these drying processes or the intense desiccation events really most relevant to the top, 10 inches? Or how far down can you go to get a, 30, 35 megapascal type drowning?
Okay, so if you look at water potential as you go down the profile, it is most, in a California annual grassland like this, it is most dry in the surface. That's a generalization. We can come back and contradict that in a minute if you'd like. But the water potential, you will, by the time you get into, so Tom Kieft was one of the early people that worked in water potential with me, and he now has spent the last, God knows how long, working in subsurface systems. And so one of the first questions we asked was, what kind of water potential do you have in subsurface systems? And they're very constant and relatively moist. I'm not talking about groundwater. I'm talking above evapotranspiration systems. So I think we actually did a meta study where we went through the literature and looked at what the average water content in evapotranspiration was, such that you're no longer impacted at all by the wetting and drying events that happen at the surface. How deep do you have to go? It's going to depend on the system. It's going to depend on the plants and their evapotranspiration, how deep they're pulling. In an annual grassland with a rooting volume that may be 30 centimeters, maybe a few roots that make it down to 50 centimeters, how deep does that extreme? Oh, actually, that one picture I had showed that. Yeah, well, actually, let's see if I can get back there quickly. Accidentally, it wasn't intentional. Well, I was going to go back to, I'm sorry, now I'm going to make you dizzy. It was field, and that's what I think we want to think about as opposed to Roman's cores, which are what happens in the greenhouse. Is there a way to go all the way back to the beginning? Okay, so if you think of that water potential in the field diagram I showed, I focused on the 4 to 10 centimeter data. But if you look at the deeper data, it shows right there. Now, so here we go. 4 to 8 centimeters is the line. I said, look at that. What happens at 45 centimeters? This is a Sierra Field Station annual grassland. These are the polka dots. So, well, they're going down. It's just they're not flashy. They're not They're not fast. They dry slowly, and they're going to stay there. Little rainfall events at the source. It's going to take a while. It's going to take a while for the wetting front to get there, if it's the wetting front that counts. If it's the wetting front that counts, okay. And also, in these environments, you have dew events on occasion in June, whenever the dew point is such. So at the very, very surface, it could be extremely dynamic, in fact, in terms of water. Rohit.
All your graphs have water potential in their axis. Can you comment on the accuracy of those measurements?
Water potential on what? Oh, like here? Oh, you don't want me to? Why don't you comment on the accuracy of those measurements? Okay, so I was waiting for you to ask, how on earth did you measure minus 30 megapascals? Is that your question? There is no instrument that can measure anywhere near that low a water potential in soil. The only way you can do it is, thank you for asking that question. I'm going to answer a different question. The only way you can do it is through what's known as isopiastic equilibration. And so what isopiastic equilibration is that a saturated solution of a known compound has a defined activity of water. And so the way we did water retention curves based on the water content of the soil, where the points on the water retention curve came from isopiastic equilibration. So the minus 35 was what? Saturated what? Sodium chloride. So a saturated solution of sodium chloride has a water activity of minus 35 megapascal. So you put a glass of that in the soil, in a closed container with the soil, let the soil sit for a long time so it comes to equilibrium, and the water content of that soil will be minus 35 megapascal. Now, the accuracy is extremely imprecise. You know that because the water content at that point is extremely low, and the change of 0.2 grams of water at that low a water content is probably going to move it from minus 33 megapascal to minus 19. So because the energetic state of the water changes extremely rapidly with desiccation at that point. So those really low numbers are very imprecise. But that they are really low, I believe. They are. Okay. Now, how about the higher water potentials? Okay. So how about the plants growing in cores instrumented with TDR that you put in? How accurate are those? Okay. Yes.
Mary, if you were so bold to try to take all of this and go to large scales with it, it struck me that water potential seems to be an enormously important explanatory variable. And I wonder, are the differences between the tree systems and the grass systems in California enough different and studied that one could say something about deep root versus shallow rooting and other critical variables that one would want to incorporate into measurements to drive models at large scale?
Okay.
Sort of several questions.
Yeah, I'm not, so I'll just answer a variety of questions. I'm not sure which ones you ask. If you could come up with reasonable data on repeatable dynamics of water potential in a system, I think it could be very useful as a parameter to drive microbial processes. Now, that's a different kind of characterization that we usually do for grasslands or forest systems. Now, frequently you'll have soil water content, okay, over time, or you have TDR. So you have TDR. and you think about it as soil, you transform it to soil water content, and now how often do you transform it to soil water potential? How often do you do water retention curves and get the energetic availability of the water? Okay, so microbes, they see energetic availability of the water, but they also see water content. Water content controls diffusion, energetic availability of the water controls the physiological impact on the cells. I would like to think that through. I think it would be fun to think about what you could do with water potential data as a driver of microbial processes, and whether you could come up with some more interesting predictions based on that. Don't know. Interesting question.
With respect to the, have you looked at under the trees versus in the open grassland? Do you see differences there?
Yes. Okay, so that's old, that's even older work. That's Louise Jackson's work, where we did a lot of work under the canopy in the open, and we measured water content, and we measured water potential. And under the canopy in a California annual grassland, under an oak canopy, which has quite an accumulation of organic material, the water content of the soil on July 1st is markedly different than the water content in the open over there. Now, the water potential is identical. When you do water retention curves on both of those soils, the energetic state of the water under the tree and in the open is the same. So the microbes are experiencing the same availability of water. The water content under the tree is higher, so diffusional supply of substrate could be a little higher. Microbial processing, if it's dependent, now that's why we want to ask, are rates dependent on energetic availability of water, or are rates dependent on diffusional supply? So that is a fundamental question. And we've got the answer for ammonium oxidation. So of course we can extrapolate that to, to everything on Earth. Sure. Yes?
Question about the TPS. When you have the wet events and the gram positives are quickly losing these small molecules, does the TPS help retain that? Can they take that back up again? Is it similar to having like an external section of life?
Interesting. You're probably right. So a lot of those pseudomonids that we saw losing their amino acids and neutral sugars, if they are in a hydrated matrix of extracellular polysaccharide. Okay, so let's say here you are in your EPS matrix. Think like a microbe. You're in your EPS matrix. Here comes the water, and 98% of your brethren die. Well, they lost all of their compatible solutes, but it's retained within the EPS. So I actually wrote a grant proposal once about nearest neighbor cannibalism, which is what it is. So some of the microbes that survive in that polysaccharide matrix would be extremely well positioned. Oh, the reviewers loved it. Would be extremely well positioned to rapidly use the carbon substrate. Yes, that's an interesting way to look at the EPS. Yeah. Any other questions? Okay. Good. I like water. It's good stuff.
Or the lack of it.
Or lack thereof, yes. All right.
Take a breath.
So it is defined fundamentally by the and so when I teach it the first time, we go start. This is what it is. You got to understand. But in a similar way.
Very bizarre.
No, I think it's really interesting because by knowing what to use there, you can start to manipulate the system to
I can't find the off.
What is
I'm
your,
looking over here. Where's the shut up? Well,
is that what you meant?
well, that would do it. Okay. Yes, thank you. There's usually a button down there.
So the transcripts, Mary, is that commonly, are people commonly making those measurements now so that we can try to use that to get at rates? I mean, are there direct, because your transcripts are just like the nitrification rates. I couldn't put them on top of each other in my head.
Oh, absolutely.
But-
When you
but
plot
they're
them, oh,
really
actually, they
tight,
do.
right?
They're very tight.
why
In
don't-
this case,
But I was going
Don't-
to ask, is this commonly the case?
okay, not necessarily.
Should we walk over and have lunch?
Sure.
Do you like it?
That 1 day time scale, or is it long term? Like the ratio between the transcriptase to the is presumably a function of the substrate.
Say it again. It's
So
presumed.
the ratio between the rates in the transcriptase, will that change from one rain event to another based on
Of course,
availability?
of course. Well, it's a function of substrate availability. The rates have to be a function of substrate availability. And a lot of people have attempted to predict rates overall based on transcripts, and it doesn't work very well.
Is it, you mean overall, like across through the whole pathway, or for individual components of it? So you were just having ammonium oxidized,
That's right, which is about as simple as you get.
right? But maybe each of the steps in that overall reaction could be done that way, and then so like DMDC, for example, a lot of these models have they calculate individual rates for ammonium oxidation, and all the things after nitrite and so forth. They're all separately calculated rates.
Okay, I think this is kind of an unusual situation where you're looking at turn on, and the rates are controlled by turn on. And so transcription tells you a lot about turn on. So microbial ecologists 8 years ago were disappointed because the number of gene copies, DNA, didn't predict anything. And then they said, oh, what we really need is the number of transcripts, because that's going to tell us what. And so then they went out and did 1 million studies looking at transcripts. And it was very disappointing. Now, in some cases, transcripts can predict. And I think if you're going to look for a case where transcripts are useful, it's in these really pulsy processes where turn on, that's what transcription is, turn on is critically important. If you're looking at an ongoing process, transcription happens here, it's totally unpredictable and hard to catch.
What's the lifetime of the enzymes?
What's the lifetime of enzymes and what's the lifetime of the transcripts themselves? And the answer is, I don't know, but it's not very long.
Hours.
Oh, I think enzymes are longer than that. Transcripts may be in hours. Depends on the enzyme, depends on the transcript.
My next question, because I forgot what I learned in your class
Yes.
2 years ago.
Yes, yes, I know. Do you remember it? Does any of that sound familiar?
What's the difference between an enzyme and a transcript?
Say again, what?
What's the difference between an enzyme? What is a transcript?
Aha. Okay.
Well, Mary, I sat in your class 15 years ago. I don't think we were even talking about it then.
Actually, I don't know that we did it then. So here you've got the DNA. And the DNA codes for ammonium oxidation, right? That's the DNA code. Now, before we can make the enzyme, we've got to come through and copy that DNA. That's called transcription. And we make a messenger RNA. That's a transcript. Oops. Now, wait, here it is. Here's the messenger RNA. It comes over here to a ribosome. And the ribosome reads the messenger RNA and actually makes tRNA, which makes the protein. So there is post-transcriptional control that can be important on occasion, sometimes a lot. So really what transcription is the translation of the genetic capacity. It's the first step in the expression of the genetic capacity.
So really you want to measure the enzymes.
Okay, so microbial ecologists said, well, what you need to know is the DNA, and that didn't work, and so we said, what you need to know is the mRNA, the transcription, and then that's not very, what we really need to know is the proteins. That's called proteomics. So that's what Jill does. And then the next question is, well, why? You don't know for sure those proteins are active. Is it post-adenylation? Why don't you just go in and do an activity assay?
Yeah.
So now
This is what you were doing
we've
20
gone
years
all
ago.
the way back around, and we're doing process assays again. Yes.
And trying to relate them to all these
Yeah, intermediate.
omics of
And it may be.
That means you feed it a substrate and see how fast it.
Yes.
So it's hilarious. You should make a comic strip out of that.
Yes, yeah. Thank you. Thank you. I assume it would be funny to you. It was kind of disappointing too. Let's see, this one is mine. And this one is, it goes in here. Here it is. No, it's part of this guy.
Well, Mary, so you know Charlie.
I know Charlie.
This is Andy Jones, who's in ERD, grad student in ERD.
Right, you look familiar, but I don't think we've met. I'm Mary.
And Zach Subin, who's
Hi, Zach.
I'm also a
I
grad
should shake your hand
student.
too, because I haven't been seen you in a while. I haven't been around. I was talking about back then.
Oh, did you?
so he's working up here now in neuroscience.
I'd be interested in hearing where you've been and what you've been doing. So yeah, you did you agree with Inez?
Yes.
Yeah, yeah. Yeah, that was, but I wouldn't have known that you had been missing. I assumed you were hiding it over there. Okay, so this will stay here, and this was the thingy that controls it.
So we're gonna ask you the same questions later about whether freezing is analogous to drying and melting is analogous to wetting.
Yeah, good questions.
Because
You want a quick answer? How about lunch?
because we're
Let's go grab a bite.
we're working on a lot of permafrost stuff.
And the answer is yes and no.
actually done work on freezing?
No,
Okay.
no, I haven't.
How about freeze drying?
Oh, we do that a lot. Well, no, not, pardon?
Just for preservation.
Yeah, just for preservation, exactly. I guess somebody else will turn that on.